Hi, my name's Rachel Pepper, and I'm a professor at the University of Puget Sound, where I teach physics and biophysics and do research at the interface of physics and biology. My field of study is biological fluid mechanics, which basically means I study how living things interact with the fluid environment around them, whether that's air or water. And this is a really interesting field because most things that are alive have to interact with either water or air to do the things necessary for that life, either feeding, reproducing, or moving around, or maybe all three. My artist collaborator was Jocelyn Curry, and it was really a pleasure and an honor to work with her. In my lab, we study two main projects at this biological and water interface. One is splash cup plants, which you can see here in a top-down view. These have these conical seed cups where a raindrop hits the cup, there's a fantastic splash, and the seeds get dispersed a really far distance, like 3 or 4 feet away from the plant. And we study the physics of this splashing dispersal, but that's not the project that Jocelyn and I worked on together. We worked on the other half of my research, which I'm about to describe to you, thinking about these very tiny microscopic organisms and how they move the water around them. So here's an example of one from our lab called Vorticella convallaria. And it's about this length of the whole organism is about the width of a human hair. So you need a microscope to look at them. They have a cell body and a stalk, and you can't see them, but they have these little appendages called cilia that they move the water with. And they're important to study because they help keep water clean. I think we can all agree that clean water is essential, both for us as humans and for the planet. But what a lot of people don't know is that these invisible microscopic organisms are one of the key things that help keep water clean and in balance. And of the microscopic organisms that are important, one really important class are these microscopic sessile suspension feeders that we study in my lab. And these organisms are special. First of all, they're tiny, but what makes them special is they live attached to the surfaces. So whether this is debris or the bottom surface of a body of water, they live attached and they're ubiquitous. They're in lakes, streams, oceans, even mud puddles. And in all these environments, what they do is they eat bacteria and debris. And because they eat bacteria and debris, they help keep the bodies of water healthy and in balance. Um, and in order to eat that bacteria and debris effectively, because they live attached and can't swim around to catch it, they draw the water towards them. So they use those cilia, the little appendages, to create a water current, a feeding current that draws the bacteria and debris towards them. And so in my lab, we study that feeding current, the physics of that feeding current, in order to figure out the feeding rate and thus the environmental impacts of these organisms. These organisms are also important, play a key role in wastewater treatment plants, and can also be important for cleanup after man-made disasters like an oil spill or a sewage leak. So understanding the feeding current and the feeding rate can sort of help understand how bodies of water can stay clean in all these different contexts. The particular study that Jocelyn and I worked on together was thinking about how organisms, Vorticella in particular, the ones we study in my lab, um, how their feeding current is changed when they're in a body of water that has flow. For instance, in a stream or the ocean, not only is there this feeding current, but there's also a background ambient flow from the body of water itself. So here's an example of one of our experiments where we studied a Vorticella feeding current combined with some background ambient flow. Here's the Vorticella in the middle, and it's attached to a surface at the bottom. And these blurs that you see are in fact beads moving through the water, very tiny microscopic beads moving through the water that have blurred because we've done a long time exposure to help us see the flow. And one of the things we discovered from this experiment is that the feeding current and the feeding rate of these Vorticella is very different depending on both the ambient flow speed and the orientation of the organism. And you can kind of see it here. As the organism, the Vorticella, shifts the orientation of its body, the feeding current changes. And we did calculations that show the way this feeding current changes drastically changes the feeding rate of the organism as well. So the big takeaway from this study was that in order to understand the feeding rate and environmental impact of these organisms, we have to understand both their typical orientations and the typical flows in the bodies of water where they are. This could also help us predict what would be the best flow to use, say, in a water treatment plant if we knew the organism's typical orientations. It was really interesting to work with Jocelyn on this project. I'd never worked with an artist having them interpret my work before. And Jocelyn brought this great both historical and visual perspective to my work to make me look at it in a little bit of a different way than I had before. And I also think both that visual and historical perspective and the way Jocelyn interpreted it, I'm hoping it will draw people in to think both about the visual, historical, and scientific sides. So I wanted to end by saying a big thank you to Jocelyn. And also thanking the other collaborators. The work that I just presented to you, I didn't do by myself. I did it with this great big team. So I'd like to thank them as well. And also thank the funders who made this work possible. And thanks to you for listening.